well, I think it's a little after 4. I just have one announcement before we start. And that's, we have 2 more weeks of seminar. Our next seminar will be Becca Durkis, and then our last seminar will be Dr. Bender. And both of those were potentially scheduled to be held in person. And so I'm just want to make sure I make the formal announcement. Everyone will get emails that both of those seminars have moved to Zoom. So all attendees will be, for the last seminars, will all be on Zoom. And I'm sure Ben will send out a reminder as we get a little bit closer. But, all right, I'll pass it over to Dr. Dillon then.
Yeah, so I am very excited to introduce Dr. Barbara Stefanska. She's an assistant professor in the Food, Nutrition, and Health program in the Faculty of Land and Food Systems at the University of British Columbia in Vancouver in Canada. So she joined UBC in July 2017 from Purdue University, and we were just talking, that's in fact where I met her. I was a graduate student in the Department of Nutrition Science when she joined as faculty. Her expertise is in the very exciting fields of nutritional epigenomics and cancer epigenetics. And Dr. Stefanska got her master's in public health and a PhD in nutritional epigenomics from the Medical University of Lodz in Poland, followed by postdoc training in the Department of Pharmacology and Therapeutics at McGill University in Montreal, Canada. So she's been to some lovely places. So Dr. Stefanska's research focuses on how diet can influence health outcomes through modulating the epigenome. And her research group investigates the link between bioactive food components, epigenetic alterations, and carcinogenesis, and establishes epigenetic biomarkers of exposure to dietary compounds as part of the exposome research. So an interesting fact about her, she was amongst the first to establish the patterns of DNA methylation and gene expression in liver cancer patients and the functional role of differential DNA methylation between tumor and normal tissue. So today she'll be presenting on the epigenetic mechanisms of anti-cancer effects of berry polyphenols. So with that, please welcome Dr. Stefanska.
Thank you, Japnat, for the nice introduction. Thank you for the invitation. I also want to say thank you to Ben for his patience with me sending all the documents that were needed. So I'm happy to be here today and to tell you a bit about what we do. So first, I will give you some background on epigenetics. I will focus mostly on DNA methylation because this is our interest in the lab, and then I will talk about some compounds we are working on polyphenols from stilbenoid class, and about some mechanisms that we see being exerted by these compounds in their anti-cancer action. So when you think about epigenetics, the simplest definition says that epigenetics refers to changes in gene expression without changes in the underlying DNA sequence. So this is the simplest definition. An excellent approach to understanding epigenetics are monozygotic twins. So we know that monozygotic twins are born with the same genetic material, and they look the same at birth. However, if you know monozygotic twins, you can say that with time, they develop some phenotypic differences. So the older they are, the easier it is to distinguish them between each other. So I want to give you a quite extreme example. So the picture over here, these 2 women are monozygotic twins. So when you look at this picture, I would say that they are siblings, but it would be very hard for me to guess that they are actually monozygotic twins. So what happened? They were separated after birth, adopted by 2 different families, and raised on 2 different continents. So really in very different environments. And you can see how the environment shaped their phenotype in a very different way. So again, they look similar, they look like siblings, but it would be very hard to say that they are monozygotic twins. So how come the same genome produces different phenotype? The explanation can lie in epigenetics. So epigenetic changes, epigenetic modifications were shown to be involved in many different disorders. And here I listed some of them. Cancer is the one that is the most studied in terms of epigenetic alterations, causal relationship, consequences, or biomarkers. So what are those? So based on what I just said, what is epigenetics and how epigenetic can impact the phenotype. So there's some kind of base that we have in our genome, but epigenetic can change the way it is expressed. So based on what I said, if you had to match these pictures in pairs in order for them to symbolize genetics versus epigenetics, what would you, for example, pair writing with? So writing you would pair with reading, but in this pair, what would symbolize genetics and what would symbolize epigenetics? So any ideas? Writing versus reading. What is epigenetics? What is genetics?
You could change the spelling of a word. Is that what you're thinking of an analogy?
So what would be the genome, so the base, and what would be the epigenome, so something that allows genome to be expressed?
Right, right.
So I would vote for A, writing as genetics and reading as epigenetics.
Yeah.
Yes, very good. And what about, so another obvious pair is musical notes, melody. So what would be here genetics and what would be epigenetics?
Come on, students.
I can see some chat.
Yeah.
So writing, yes, so reading is epigenetics, melody epigenetics, yes. So musical notes is genetics, melody is epigenetics. So almost musical notes, when you see musical notes, it means practically nothing if you cannot translate it into melody. What about computer and software, the last pair?
Software genetics?
Okay.
I think software is epigenetics as it translates what the computer's doing.
Yes, yes, exactly. So a computer itself is just a piece of metal. If you open it and you don't have a proper software, you cannot even write a letter, you cannot prepare a PowerPoint presentation, for example. So computer itself is just, is genetics, is some kind of base that we need. But in order to really, for computer to perform its function, we need software. So when you have trouble in understanding what is epigenetics, I really want you to come back to these pairs and try to think about writing versus reading. Writing is just some base, so it's the same with genetic code. It's just some sequence. It practically means nothing if it cannot be expressed. And what regulates this expression? Epigenetics. So what is important about epigenetics is the fact that it's quite dynamic, although can be also passed on to next generations. It's dynamic, so it means that it is affected by many factors, including many environmental factors, like, for example, diet. So in this slide, you can see three basic components of the epigenome: DNA methylation, histone covalent modifications, and non-coding RNA mechanisms. So today I will go more into detail in terms of DNA methylation. And these three components are the basic components. Now, today, we know that it's not everything there. there's something additional that complicates the whole epigenome. So recent 10 years of research show that there are additional proteins, sometimes even complexes of proteins that modify chromatin structure. Chromatin is DNA wrapped around histones. And as you can see here, this histone octamers with DNA can be very close to each other, and this is compacted chromatin, or they can be quite far from each other, and this is open chromatin. So in open chromatin, we would expect that transcription should happen. So genes should be expressed, we should have a protein produced based on our genome. However, sometimes there's this big complex sitting, for example, polycomp-repressive complex, that just covers the region. And although there is no methylation, DNA methylation, histone marks are active, the region will be repressed. The transcription won't be possible to happen because this additional complex, chromatin modifying complex, sits there and covers the sequence. So these are the consequences of all these epigenetic marks. In addition, all these components, so now we would say it's DNA methylation, histone modifications, non-coding RNA mechanisms, and chromatin modifying complexes. So all this is affected by many environmental factors. And you can see chemicals, viral infections, radiation, temperature, even social economic status, abusive behavior, even maternal care, and of course, diet. So I will focus today on diet because this is our interest in the lab, but you can imagine how many factors can actually impact the epigenome, and consequently expression of our genes, level of proteins in our body, and as a result functions. So DNA methylation, this will be our focus today. It's a reaction synthesized, catalyzed by DNMTs. These are enzymes, DNA methyltransferases. These enzymes takes a methyl group from SAM is a methyl donor, and they just transfer this methyl group into 5th position of the cytosine ring over here. And as a result, we have 5-methyl cytosine. You can see in the sequence that this methyl group will stick out. In some way, it will stick out. So when it sticks out, we can expect that the binding of different proteins to this region will be affected. So how DNA methylation can regulate gene transcription? So over here, you can see visually how this happens. So this is promoter region, and all these green lines represent non-methylated DNA. So non-methylated cytosines. And you have transcription factors binding, RNA polymerase II binding, and transcription is possible to happen. Gene is expressed, we have a protein. So this is a promoter. This is an example of an enhancer. Enhancers are fragments that are quite far from the beginning of the gene, so from the transcription start site. When these cytosines are methylated, so here marked in red, other proteins are recruited, so-called methyl binding domain proteins. They recruit, in addition, DNMTs that will even more methylate the sequence more, and the whole region is practically masked. Transcription factors cannot see it, cannot recognize, because it's covered with all the other proteins. And transcription is not possible to happen. The gene is not expressed, and we don't have a protein, or we have low levels of a given protein in our body. So in general, when regulatory regions of genes like promoters, enhancers, lose methylation, so there is decrease in methylation, hypomethylation, we have activation. So there's a reverse relationship between the level of methylation in these regions and transcriptional activity. If these regions gain methylation, so we have hypermethylation, we will have silencing. More methylated region, regulatory region, means silencing. So these are regulatory regions. The situation with gene bodies is a bit different, and I won't go into it today because we will, in our studies, we focus mostly on promoters and enhancers. So in cancer, we know now that the patterns of DNA methylation change, and they really change drastically. New technologies like microarrays, sequencing, next generation sequencing gave us the opportunity to see really in detail what's happening. So over here, you can see an example of a heat map on a heat map showing genome-wide analysis of DNA methylation by microarrays. So all these rows here correspond to genes, and over here we have different samples from patients. This is normal tissue, and over here we have tumor tissue. And you can see that there is a subset of genes that are methylated in normal, so here marked in red, and they actually lose methylation in cancer. So you see that you have more green, lower levels of methylation. On the other hand, there's also a subset of genes that are that is non-methylated or lowly methylated in normal, so green, and it gains methylation in cancer. So we have more red. So for some subsets of genes, the pattern of DNA methylation is practically reversed in cancer. So what are those genes? Studies showed, and many years of studies showed, that tumor suppressor genes, so those that are some kind of guardians in our body, so they protect us from developing cancer, they are non-methylated and expressed in normal tissue. However, in cancer, for some reasons, they gain methylation and they get silenced. So we lose these genes. On the other hand, oncogenes is another group of genes that are methylated in normal tissue and non-expressed, and this is good. The oncogenes, oncogenes usually are genes that increase proliferation, increase migration, so increase function that are associated with cancer development. And we actually express these genes, but only when we need it. So there are some periods in our life when we need these genes and we will express them. Otherwise, we want them to be silent. So what happens in cancer development? These genes lose methylation, DNA methylation, and become expressed. So we have almost two forces coming together. On one side, we lose tumor suppressor genes. On the other side, we gain oncogenes. And together, this consequently leads to cancer. We still don't know. There are some changes that are associated with initiation. Other changes may be a consequence of cancer development. So it's more complicated, but we know now that there are definitely some changes that are associated with initiation of cancer. So in terms of tumor suppressor genes, we have quite a lot of pieces of evidence showing that bioactive compounds, and here we have, I listed a few of them, they actually reverse methylation of tumor suppressors. And this can be one of the mechanisms of anti-cancer action of these compounds. However, how these compounds affect oncogenes and epigenetic regulation of oncogenes is really not known. So in my lab, we want to know if, first of all, if oncogenes are regulated epigenetically, and second thing, if bioactive compounds from foods, including polyphenols from berries, grapes, blueberries, can change, reverse the altered pattern of DNA methylation within oncogenes. So some studies show that there is an effect of bioactive compounds on expression of oncogenes. However, really mechanistic studies on how expression is affected and why, it's not addressed. So this is our main interest in the lab, next to many other things, but today I will focus on this. So first, what we wanted to test if bioactive compounds from berries, like for example PTS, pterostilbene, affects the phenotype of cancer cells. We focus on breast cancer. MCF10CA1A cells are highly invasive, and we treated these cells with PTS. We saw very nicely decrease in cell growth. At the same time, we used a normal cell model like MCF10A and tested also the impact of the compounds on cell growth. We were aiming to establish a dose that will decrease proliferation of cancer cells without high impact on normal cells. So we want to really affect cancer cells with minimal effects on normal cells. So this dose, we called it IC50, so decreasing cell proliferation by 50% compared to control. And we look also at viability. So we wanted to make sure that at these doses, the cells will be viable. So we don't want to use high doses to kill the cells. We want to use low doses to change cells at the level of molecular makeup, and this way prevent or decrease cancer development and cancerous properties. So we established these doses and we use them in subsequent studies. So cell proliferation was affected, but also invasion. So we tested, you can see over here, this is control, this is resveratrol, RSV, this is pterostilbene, PTS. So two compounds from berries, and they decrease invasion of cells. 1H is breast cancer non-invasive, 1A is breast cancer invasive, and this is normal cell model. So you can see nicely that invasion of cells decreases. So the ability of cells to invade through extracellular matrix is lower. So this is actually what we want to see if a given compound has anti-cancer properties. Also, soft agar assay show nice decrease. Soft agar assay refers to anchorage independent growth. So normal cells, if you have one single normal cell, it's really very hard for the cell to divide. The normal cell cannot form colonies from one single cell. It needs contact with other cells, but cancer cells are so potent that they actually can create colonies from one single cell. And if they can do that, it means that they really have high cancerous properties. So we can see that the compounds decrease the ability of cancer cells to form colonies from one single cell. So they cannot grow in soft agar as well. Soft agar decreases the contact between cells. So I will divide results now into three parts. In part one, we will address our hypothesis. So we hypothesize that these phenotypic differences, we hypothesize that the effect of the compounds on the phenotype is associated with changes in the epigenome. And our interest was DNA methylation. So we performed first genome-wide analysis of DNA methylation using microarrays. You can see that in non-invasive and invasive cells, we have a lot of changes. So each bar going up or down corresponds to change in the methylation level of a single CPG site. So we have many CPGs affected, many sites. The red one corresponds to increase in DNA methylation. The blue one corresponds to decrease. Obviously, here from these pictures, you can see that we have more red. So more CPGs that actually gained DNA methylation. So we were interested in this group of genes, these changes, and also we hypothesized that maybe among those genes that gain methylation, we have oncogenes. This would make sense based on our previous studies that oncogenes gain methylation and become silenced in response to polyphenols. So we look more into detail at these genes. One of the highest changes, so a CPG site that gain methylation at the highest level was located in a gene called MAML2. And it happened that this gene is an activator of the notch signaling. Notch signaling is one of the oncogenic pathways, so pathways that drive cancer development, at least breast cancer development. There are some differences depending on cancer type. So we were interested in this gene. We performed several mechanistic studies. So first of all, we wanted to confirm if there is actually increase in DNA methylation. So we used another method, quantitative method, pyrosequencing, and we confirmed that there's increase in DNA methylation upon resveratrol and also pterostilbene. And this increase in DNA methylation corresponded with reduced expression. So it made sense. The gene is methylated and less expressed in response to our compounds. So we followed up with more mechanistic studies, and I won't present these studies now. They are already published, and today I want to focus on more exciting things that are not published yet. Changes in MAML2 were associated with inhibition of notch signaling, which also made sense. So these are three genes that are notch signaling related genes. MAML2 was downregulated, and this corresponded to downregulation of these genes. So it means that the pathway is inhibited. So the change in MAML2 that we saw had biological consequences, which is inhibition of notch signaling. So in general, if MAML2 is expressed, it creates an activator complex which activates transcription of notch target genes, and this drives cancer development. However, upon polyphenols, MAML2 gets silenced because of DNA methylation, and repressor complex is formed. This repressor complex won't drive transcription of notch target genes, and eventually cancer development progression will be inhibited. So this was our model that we mechanistically studied. So what we found is that there is a transcription factor, OCT1, that drives transcription of MAML2 in cancer cells. Upon polyphenol treatment, something happens. OCT1 disappears and DNMT3B is recruited to this region. Recruitment of DNMT3B causes DNA methylation and silencing of MAML2. So this was our final conclusion from mechanistic studies, and this gave us a reason to form a new hypothesis, and I will come back to it in a moment. So you could ask me, okay, so you study all these things and you see inhibition of notch signaling, but is it really significant? So it is. One of the struggles in cancer therapy is resistance to therapy that develops with time, and also side effects. So there are existing inhibitors of notch signaling, like gamma secretase inhibitors. They inhibit the pathway at this level over here. And we have side effects, we have resistance to therapy. So with polyphenols as an alternative approach, we could combine the treatment and hopefully be able to use lower doses of gamma secretase inhibitors, have less side effects, and decrease the chance of developing resistance to therapy. So this is something we want to also explore in the future. So what we see, it is significant, and it may be, it may have future applications in the clinical settings. So our next question was, okay, we see all these changes, but it's in vitro. Okay, we have 2 cell lines, but it's still in vitro. So is it really happening in vivo? So we looked at in vivo model. It was a rat study with another cancer type. We looked at liver cancer. Liver cancer is one of the types that is really very affected by environmental factors. And it is known that there are many changes in DNA methylation happening in liver cancer development and progression. So we had 4 groups of animals, and we used a methyl donor deficient diet to trigger liver cancer. We supplemented this diet with resveratrol and pterostilbene. We wanted to see what will happen with cancer development. So in general, what we observed first, like our first finding, was that there's a decrease in cancer in the number of nodules. So CSAA, it's a normal, healthy diet. CDAA is a diet that triggers cancer. CDAA supplemented with pterostilbene. When you look at this pictures from histology, you can see that over here we have a lot of lipid droplets because fatty liver cirrhosis, fatty liver is one of the elements of liver cancer development. And you can see that after supplementation, the histology really resemble the healthy tissue. And the number of nodules goes down. Pterostilbene was much more effective compared to resveratrol. So we went further to explore molecularly what happens upon pterostilbene supplementation. So when we compare tumors versus normal, we saw increase in notch signaling. And one of the genes that was really upregulated strongly was MAML2 actually. What happened when we trigger cancer, MAML2 lost methylation and got expressed. When we supplemented with pterostilbene, methylation went up, and this corresponded with downregulation of the gene. So very nicely, we observed similar effects as in vitro. So this was really important for us. We saw that really it wasn't some artifact or something. We have another cancer model, we have in vivo model, and we observed similar mechanisms. Notch target genes were nicely down-regulated after supplementation of the diet with pterostilbene. So again, similar effects to what we saw in vitro. So now I will move to the last part of the story. So I said that this is what came out from our initial studies. So we think that there's OCT1 sitting and driving expression of MAML2. Polyphenols change binding of OCT1, and instead of OCT1, DNMT3B starts binding to this region, methylates this region, and MAML2 get silenced. So the question arose from this findings, if DNMT3B really is responsible for methylation of this region. It may bind there, but maybe it's not crucial. So first we performed a CHIP, so chromatin immunoprecipitation. We wanted to find regions where OCT1 binding changes in response to our compounds. In these studies, we used pterostilbene. Pterostilbene has higher bioavailability compared to resveratrol, so it seems to be more interesting. So we focus on pterostilbene. So OCT1 binding. You can see that we found a lot of regions where OCT1 binding actually decreases in response to pterostilbene. So this would make sense. So we wanted to know more about these regions. If we hypothesize that OCT1 drives expression of oncogenes, and when we treat with polyphenols, expression of oncogenes goes down, it would also indicate that OCT1 binding should go down, because it's a transcription factor that drives expression. So binding of OCT1 at oncogenes should go down. So our hypothesis was that among those regions over here, we have oncogenes. So regions within oncogenes. We looked where this binding changes in binding happens. So some portion was in promoter and enhancers, because promoter enhancers are known regulatory regions of genes. We focus on these genes in our further studies. So OCT1 binding, we know now a bit more about where OCT1 binding changes. However, there was another part of the story, DNMT3B. So we performed independently a CHIP with DNMT3B in order to find regions where DNMT3B binding changes in response to polyphenols. So in this graph, what I wanted to say through showing you this graph is that there are certain regions where OCT1 reduced binding of OCT1 corresponds to increased binding of DNMT3B. And practically, these are the regions that we want to explore. From our initial studies, it looks like OCT1 drives transcription of MAML2. When we treat with polyphenols, OCT1 disappears and DNMT3B binds. So these are the regions, this 47 unique genes contained regions where OCT1 disappeared and DNMT3B came to bind. And this really happened at the same region. So we were really looking to find the real overlap. So not somewhere in the same gene, but exactly in the same region. So we look more at these genes. There were many, many interesting genes. I listed here some of them. Two of the genes contained two fragments with OCT1 reduced binding and DNMT3B increased binding. They were in promoter and enhancer. So we focus on one of the genes, PRKCA. PRKCA is a kinase that regulates many oncogenic pathways. So quite interesting gene. So we looked more in detail. Over here, you can see tracks with changes in binding of DNMT3B and OCT1. So over here, you have a region. So this what is pink. It means that we have more binding of DNMT3B in response to polyphenols. This what is blue, it means that we have less binding in response to polyphenols. So you can nicely see that a region that gained DNMT3B lost OCT1. So pink and blue. So this is the region that we explored more in detail. If OCT1 disappears and DNMT3B binds, it would mean that there should be DNA methylation. First of all, the gene should be silenced. So let's look what happens. So first of all, we used a quantitative method to check binding one more time. So we didn't rely only on sequencing, but we performed Q-chip, so quantitative chip analysis. And you can see that OCT1 binding goes down very nicely and DNMT3 binding goes up. This corresponds with increase in methylation. Here you have a region containing 9 CPG sites, and many of them very nicely go up. And expression of the gene goes down. So this is what we hypothesize, and we confirm that for PRKCA, it looks like DNMT3B could be involved in methylation. So is it really? We used CRISPR technology to knock out DNMT3B in breast cancer cells. So over here you can see a confirmation of knockout. So DNMT3B is not expressed. Interestingly, we saw that upon knockout, pterostilbene very nicely impacts proliferation of the cells. So when we treat the cells with pterostilbene, cell growth went down. So there is an effect of pterostilbene on knockout cells, but now it was interesting for us to see if this effect will be associated with PRKCA hypermethylation. And as you can see here, there's no hypermethylation. So it means that if we remove DNMT3B, methylation doesn't happen in response to pterostilbene. So we proved that hypermethylation that is observed in PRKCA in response to polyphenols is DNMT3B dependent. So this we proved using our model and CRISPR technology. However, interestingly, expression of PRKCA upon treatment with pterostilbene in knockout cells was still down. So although there was no hypermethylation, expression was down. So we confirmed that hypermethylation is DNMT3B dependent, but clearly the studies show that there is another mechanism that pterostilbene triggers to decrease PRKCA expression despite lack of DNA methylation. So maybe there are histone modification that change, and transcription will be active. Transcription will be silenced, although there's no hypermethylation. Another possibility is that there is some regulatory mechanism at the protein level. So again, a very interesting observation, and this decrease in PRKCA expression can also explain the previous slide that growth of cells goes down when we treat with PTS. So PTS maintains its effect, but regulates PRKCA not through DNA methylation anymore, because DNMT3B is gone. So in cells without knockout, hypermethylation that we observed, it looks like it is DNMT3B dependent. So just to conclude, we see that the epigenome is dynamic. It is affected by many factors, including our diet. And it is, so we study bioactive compounds that are supposed to maintain health, prevent cancer, prevent disease. So it's a very, very good news. It's a very good news, because we can change the epigenome using diet in order to maintain our health. And the mechanisms are quite complicated, but our studies show that DNMT3B could be important for bioactive compounds to exert the effect on DNA methylation. So thank you very much for your attention. I would be happy to take any questions. I hope that everyone stayed with me and I didn't lose anyone with a quite complicated topic. Thank you.
Thank you so much, Dr. Stefanska. So before we start questions, which I see a few starting to pop in now, thank you. If we want to give her some applause in whatever way you like, whether if it's on screen or with the emoticons. Yes. So I will, everyone runs the Q&A facilitation a little bit differently. So here is my plan. Of course, you're welcome to use the chat, and then I am going to keep an eye on the participants. So if you're more comfortable with raising hands, then I'll keep my eye there and then can call on you. It's my teacher role coming out. So if you'd like to do that. So first, I'll go ahead and start with what one of our dietetic students has there in the chat, which is looking at the different models. And she actually has almost the exact same question that I had, in case that I needed an icebreaker question. Is as we are translators of nutrition science to the public, if you have an idea about what the food dose would be to attain the dose you're seeing those effects with, we would very much like to hear more about that.
Yeah. So in terms of, for example, so the doses that we use in our studies are close to physiological doses. So we use in vitro, we use a prolonged cell culture, 9 days, in order to really decrease the dose and still achieve a phenotypic effect. In terms of translation and how many berries cancer patient, for example, needs to eat. So I would say the bioactive compounds would be very important for prevention. And then we can talk about consumption, dietary intake, and prevention. However, if it comes to cancer patients, I wouldn't talk about dietary intake, because obviously the doses that we need to use are higher than what we can achieve from the diet. So it would go into more therapeutic applications, and we can purify the compound and have a pill, for example. So when we talk about combining polyphenols with gamma secretase inhibitors to inhibit the notch signaling pathway, we would go for sure for a pill instead of a dietary intake. So the doses for therapeutic applications would be higher compared to what we can achieve from the diet. In terms of prevention, in terms of prevention, studies show that what is important is chronic exposure. So by eating, for example, berries on everyday basis, we practically achieve some kind of cumulative effect. And although the doses are very small, because it happens on everyday basis, the effect will be visible and will be exerted. So it's always very hard to translate what we do in the lab. And unless you study people, it's hard to translate the doses. In our animal studies, we use quite high doses. It was cancer, so we were looking more at therapeutic rather than prevention. In vitro cell lines, we really used doses that were very close to peak plasma concentration. So for example, for resveratrol, peak plasma concentration is around 3 micromolar. And in our studies, we used 15 micromolar. So just a bit higher, but very close. Yeah.
Thank you. Keeping an eye out to see if there's any other hands raised.
So how absorbable are the compounds? Can you count on supplementation strategies? So in terms of bioavailability, polyphenols in general, we know now more and more. So I will give you an example of genistein. So we know that genistein, upon getting into our system, is metabolized in intestinal cells and in the liver through phase 2, and then gets into the main circulation, enters cells in peripheral tissues, and actually inside the cells, these metabolites are converted back to genistein. So this is really interesting and really encouraging. So in our studies, we used purified compounds, parent compounds, not metabolites. Studying metabolites will be also very important. So only 10% of ingested polyphenols gets into phase 2 and enters blood circulation. 90% is metabolized by gut microbiota. So they are not lost. These metabolites that are produced in gut microbiota will get into our main circulation, will impact the cells. So at the end, if you talk about bioavailability, you focus on the parent compound. So you would say that it's low. But at the end, through metabolites, there may be a very, very strong effect and high bioavailability at the end.
I have one, so if you're got one that's you're waiting to type, then please go right ahead. Like I said, I am watching for hands in all the different ways people can raise hands. But one, I actually have a question about methodology with the animal studies that you had with that diet. Can you talk a little bit more about how the animals received that diet? So did they get the cancer promoting diet for a certain amount of time and then have the compound, or was it a part of that? It kind of gets at that translational, who are we targeting here?
Yeah. So it was a pilot study, and in Fisher 344 rats, to trigger liver cancer, fully developed tumors, you need to maintain the rats on methyl donor deficient diet for 1 year. And I wanted to do that because it was a pilot study. I really wanted to do what will happen at the end. So now in follow-up studies, I would do several groups and different exposure time. But in this pilot study, we we applied methyl donor deficient diet for 1 year. And we supplemented the diet with polyphenols throughout the whole year. We started supplementation 3 weeks earlier compared to methyl donor deficient diet. So it was some kind of a pre-exposure, and then we triggered liver cancer. We triggered liver cancer through methyl donor deficient diet.
Thank you so much.
Do you think that anti-cancer effect of these compounds in berries would vary depending on the type of cancer in question? Thank you, Trey. So I would say that yes, it could. The effects that we see in breast cancer and liver cancer are quite encouraging. So it looks like this mechanism of inhibition of the Notch signaling pathway is happening maybe in different types of cancer. It's not only in one type of cancer, but in general, we see differences in different cancers. Even in terms of Notch signaling, in some cancers, actually, Notch signaling is acting as a tumor suppressor pathway, not oncogenic. So it's even like more complicated. So I think that it also depends what kind of changes are in a given cancer, and then the compounds will exert anti-cancer effects in a bit different way, and probably affect different mechanisms.
I have a question. I could just ask directly.
Yeah.
I was wondering, in absence of chronic exposure, how would these would there still be positive effects of dietary polyphenols and epigenetic changes in older adults?
Yeah. So you mean, like, let's say we eat, and if we get changes in the epigenome?
Yes,
just upon one exposure.
Hasn't been consuming these polyphenols, as growing up. By the time they reach old age, would changing their diet at that point, would we still see these beneficial changes?
So I think that yes. So at any point in your life, if you want to change the diet to a healthy patterns, it's for sure, it will be beneficial, for sure. No matter at which point in your life you are. The question can be, if you are still healthy, so dietary patterns will have benefits for prevention. If you are already with a condition, especially like cancer, it could be a bit more complicated. So bioactive compounds work a lot as antioxidants. Depending at which stage of cancer you are, this antioxidant activity can sometimes help cancer actually to grow instead of fighting cancer. It also depends on the doses. So I always say, what we study is you need to think more about prevention. I wouldn't say that you can take these compounds and use to treat people. You can use to support existing therapies, and this is good, but I wouldn't say that you can use them to treat people, because they have very broad spectrum of action. When you have cancer, when an individual has cancer, we need to act fast. So we need to have something really potent, usually targeting one thing and very potent. So a drug. The compounds we can use to support it, but I wouldn't say to treat. So when you are healthy and you want to change your diet, it will be always beneficial. If you have already a condition, then it, and especially when you are on different drugs, then really it may be a bit more complicated. We also see very interesting observations, recent observations in our lab. We see that the effects exerted by the compounds at the epigenome level, it looks like we need to keep exposure on in order to maintain these effects for a longer period of time. Which makes sense. These compounds are not toxic. We use low doses. They change the epigenome, but they need to be there in order to maintain these changes. When we withdraw them, then the epigenome is dynamic. So the changes, alterations may come back. So very interesting observation, and we are still looking into it.
I want to be good stewards of everyone's time, so I want to offer one last opportunity before we wrap up for today. All right. Thank you so much, Dr. Stefanska. This was wonderful. I really enjoyed your presentation. I know very translational for our people in the audience, so we very much appreciate that. If we could, in whatever form is best for you, if we could offer some thanks and gratitude by applause or the applause emoticon, we appreciate it very much. Thank you.
And yeah, it was a pleasure to be with you today and to share our work. Thank you so much. Thank you, and thank you for the invitation.
Yeah, thank you.
It's nice. I was laughing to my colleagues. Oh, in half an hour I will be at University of Missouri. So we have this opportunity thanks to all these online tools.
Yeah, exactly. That's what we were discussing too. We're just excited to have you from Canada.
Yeah, exactly. Thank you so much. And yeah, hopefully in the future we'll be able to meet in person at some point.
Yeah, while people are leaving though, I did have a follow-up question that is definitely more personal than all this. I do, one of the courses I teach is lifespan nutrition here, and we do talk about epigenetics and like generational health sort of things. And as you were answering Dr. Dillon's question there with how these particular compounds need to be present, you know, we need to have like a kind of a supply of it. We had a wonderful talk a few years ago here, You Are What Your Grandparents Ate. And it was really talking about that, that epigenetic modifications that you may have. We think that all of them fall off, but not all of them do. So I would be interested to hear what your take is on that. And if you think that there are certain compounds or certain parts, and we can certainly, I'm happy to entertain this via email. I can do that because I know you're busy and it's past 5. And
That's okay.
Other people also, but I would be interested if you, what your take on that idea is, You Are What Your Grandparents Ate, and its effect on health.
Yeah. So epigenetics is dynamic, but at the same time, it can be passed onto next generations. So there are some changes that will persist, and these changes will be passed on to next generations. So what our grandparents ate will matter. What our parents ate will matter. And we know now that it's not only about a mother and her body, but also about the father, because changes in sperm DNA may have also impact for the offsprings. So yeah, really so-called transgenerational epigenetics is a very hot topic, very interesting, complicated, but yeah, we know that. Yeah, I was talking. when I talk about how epigenetic changes can be transferred into next generations, I always give the example of a study where they used a smell of acetophenone. And there was, when animals smell acetophenone, they were given electric shock in their foot. So it was some kind of stress. And then babies, only upon acetophenone, without any electric shock, they were stressed. They were reacting in a way like almost they got electric shock. So very, very interesting field.
Thank you so much, and especially for entertaining a question past time. I appreciate it quite a bit.
Thank you. Thank you so much, and stay well.
You as well.
Yeah. Thank you. Thank you for the invitation.
Thank you for coming. I mean, you know, virtually as well.
Yeah, I know.
Bye.
Thank you so
Bye.
much. Bye-bye. Thank you.